And so our next presenter is going to be Dr. Gilbert Gallardo, also from the Washington University School of Medicine in St. Louis. And his project is to identify small molecules that suppress astrocytic-dependent inflammation and evaluate the role of mTOR in autophagia signaling in reactive astrocytes. If you don't understand any of that, I'm sure he will do well in explaining it now. Dr. Gallardo.
Okay, well, thank you so much for this opportunity to share our proposed studies, and I'm excited to be working with the foundation for the next couple of years. And I will try to share my screen here. Okay, so my name is Gilbert Gallardo, and I am an assistant professor in the Department of Neurology at Washington University School of Medicine, where my lab focuses on trying to understand the contribution of astrocytes on progressing Alzheimer's disease. And the goal of this proposed studies is to develop anti-inflammatory molecules that can prevent the inflammatory response. And we've heard some really nice backgrounds on Alzheimer's disease, and this slide kind of recapitulates or summarizes some of the things we've been hearing. And it's commonly thought that Alzheimer's disease is initiated by a deposition of amyloid beta plaques that are extracellular. And amyloid plaques are composed of these amyloid beta peptides that are synthesized by processing the amyloid precursor protein that we just heard. Now, shortly after the plaques are deposited, the second component of the disease process is an inflammatory response. Now, the inflammatory response is composed of both the astrocytes and the microglia that surround these plaques. Now, the actual definition of neuroinflammation is still really not clear because we really do not understand what inflammation is doing. We know that's an early process, and some initial studies are starting to suggest that early on, this inflammatory response may be protective. But what we do know is that as the disease progresses, this inflammatory response becomes detrimental, and that the reactive astrocytes and the microglia tend to progress the neurodegenerative process together with tau pathology. Now, we heard about tau protein, and tau is normally confined in the axons of the neurons, but as a disease progresses, it starts to be hyperphosphorylate and then liberates itself from the axons and starts to form hyperphosphorylated neurofibril tau tangles. Now, in Alzheimer's patients, there is a clear association with the accumulation of neurofibril tangles and also with the inflammatory response, with the progression of dementia and also the degenerative process. Therefore, it's commonly more thought that the plaque deposition is an early event, whereas the progression is more related to the inflammatory response and the accumulation of tau pathology. So understanding the contribution of astrocytes and microglia together with the tau pathology may enable us to develop some therapeutic interventions. Now, although we do know the inflammatory response is a component, the mechanisms of how astrocytes or microglia regulate the inflammatory response is really not known. Now, a couple of years ago, we actually found a molecule in astrocytes that regulates the astrocytic response. And this molecule is the alpha-2 sodium ATPase, which controls the ionic homeostasis. And in astrocytes, in a mouse model for ALS or amyotrophic lateral sclerosis that also displays neuroinflammation and motor neuron degeneration, we found that this alpha-2 was elevated. And we hypothesized that if we decrease or knock down the expression of this alpha-2, we can potentially protect the loss of motor neurons in our mouse model for ALS. And in doing so, we did a genetic cross in which we can decrease the amount of expression of the alpha-2, and we found that indeed, this led to an increase in survival of our ALS mouse model. Typically, these mice die off for around 160 days. Decreasing the expression of the alpha-2 increased their overall survival. So this initial study suggested that one mechanism by which astrocytes regulate an inflammatory response is by regulating an upregulation of the alpha-2 sodium ATPase. And our next question was, is it possible that the astrocytic alpha-2 sodium ATPase also plays a role in Alzheimer's disease? Now, this study here is the foundation of our proposed studies. We first looked at the levels of sodium ATPase in human AD or human Alzheimer's patients, that's postmortemly. And we found that the expression of the sodium ATPase is elevated, very characteristic to what we found in ALS. And taking a mouse model that displays tau pathology, we found that the sodium ATPases age-dependently increase, together with astrogliosis. This marker here is GFAP, which is a marker for reactive astrocytes, and it correlated with the abundance amount of alpha-2 sodium ATPase. Now, using this mouse model that develops tau pathology, which is evident here, this is a stain that recognizes pathological tau. And this accumulation of pathological tau is age-dependent. We decided to block the activity of the sodium ATPase using digoxin. Now, digoxin has commonly been used as a drug for hypertension and is selective for the sodium ATPase. However, it is impermeable to the brain. So we actually infuse this drug directly into the brain for about 2 months and measured the amount of tau pathology. And we found that infusion of digoxin, which blocks the activity of sodium ATPase, significantly prevented the accumulation of tau pathology throughout the hippocampus, in which Joe had mentioned. The hippocampus is an area for learning and memory. And this is just a measurement in that after infusion of digoxin, we prevented the accumulation of tau pathology. Equally important, these mice also display significant brain atrophy, and there's enlargement of the ventricles and thinning of the cortex. Infusion of digoxin also blocked the brain atrophy. So this initial study suggested that if we target the sodium ATPase, it may be beneficial. However, there are some limitations. First of all, digoxin is not penetrable to the brain, so we cannot use this as a therapeutic intervention. The second limitation is that we still don't understand how the alpha-2 is actually regulating an inflammatory response. Therefore, the first aim of the proposed studies is to identify new molecules that could potentially block the sodium ATPase that display brain penetrance in collaboration with the Center for Drug Discovery here at Washington University School of Medicine. And how we aim to do this is by utilizing the sodium ATPase microplate activity assay, which is commercially available. We can now plate astrocytes in a 96-well plate and add small molecules that have been predefined to penetrate the brain and try to identify molecules that block the activity by utilizing the sodium ATPase microplate assay. Candidate molecules will next be screened in a second assay, in which we have developed this cell culture model for Alzheimer's disease. In a collaborative study a couple of years ago, what we found is that astrocytes that express the ApoE4 allele. Now, ApoE4 is the highest genetic risk factor for developing Alzheimer's disease. And we found that astrocytes that express ApoE display neurotoxicity. And we can show this neurotoxicity by taking astrocytes and putting them on a plate. And on top of that, you put neurons on. Now, this is our control. And we see that these neurons are relatively happy. However, when we plate astrocytes that express the ApoE4 allele, these neurons tend to are rather unhealthy, and about 14 days later, they begin to degenerate. Now, we know that this toxicity is dependent on the sodium ATPase, and that if we knock down the sodium ATPase in E4 expressing astrocytes or ApoE4 expressing astrocytes, put neurons on top of that, we now protect the neurotoxicity. So the second screen will be to identify small molecules that decrease the activity by using the sodium ATPase microplate assay. And the next screen will be used to see, do these small molecules prevent the toxicity of ApoE4 expressing astrocytes? Long-term would be to develop these molecules to see if we can prevent the neurotoxicity and the tau pathology in our tau transgenic mice. The next question is to try to identify a mechanism by which the sodium ATPase regulates inflammation. Now, as I mentioned, the sodium ATPase is an ionic pump. It regulates the sodium and potassium by transporting the ions across the cell membrane, and it utilizes ATP. In the brain, the sodium ATPase are the largest consumers of ATP, and we hypothesize that the increased elevation of the sodium ATPase may alter the ATP levels or increase metabolism. And one molecule that is a sensor for the metabolic stress or metabolisms of cells is AMP-kinase. AMP-kinase measures the amount of ADP to ATP ratios. Upon increased metabolic rate, we get an increased amount of ADP. This activates AMP kinase by phosphorylation. Now, AMP kinase is a signaling molecule that can regulate a number of signaling events. Most notably is an mTOR and autophagy pathway. Now, mTOR and autophagy go hand in hand. mTOR inhibits autophagy and autophagy inhibits mTOR. These mechanism is a mechanism by which how cells regulate metabolism. And we believe, or we hypothesize, one way of how chronic inflammation is occurring in Alzheimer's disease is by a dysregulation of the sodium ATPase, which leads to a decrease in ATP levels, activation of the AMP kinase, which then would dysregulate the mTOR signaling and increasing autophagy. Now, autophagy is a mechanism in which how cells regulate or degrade components within the cell. Now, we do have some preliminary data to suggest that astrocytes, when activated, do regulate the AMP kinase pathway. We use these cultured astrocytes in which we stimulate them with LPS, or looking at these ApoE4 astrocytes. And we can see that upon stimulation, there is an increased phosphorylation of AMP kinase, together with this astrogliosis marker. Now, this LC3 is autophagy protein, in which an increase in autophagy leads to a degradation. So we see a decrease in this LC3 protein. Another arm of the mTOR signaling is this AKT. Upon phosphorylation, it activates mTOR signaling. And we see that after we activate astrocytes, we see that there's a decrease in phosphorylation, suggesting that indeed one mechanism by which astrocytes are regulating an inflammatory response may be through this AMP kinase pathway, in which it decreases the mTOR signaling, but increases the autophagy pathway. So our goal is to better understand this mechanism, and that if we can identify signaling molecules that are downstream, we can now have new therapeutic targets for preventing the inflammatory response and hopefully prevent the degenerative process in Alzheimer's disease. So our goals are to identify new inhibitors of the alpha-2 sodium ATPase that penetrate the brain, and also understand the mTOR autophagy signaling mechanisms in astrocytes, with the idea that we can identify new molecules for potential therapeutic intervention. With that, I'd like to say thank you to obviously everyone here and also the CART Trust Fund. In the lab, Carolyn Mann developed the digoxin study. The study is under revisions now, and a number of people are now following up on this study. Collaboration, the Center for Drug Discovery will be providing the small molecules. They have well over 50,000 small molecules that we can actually screen, and other funding agencies, and also my moral support.
Thank you so much, Dr. Gallardo. Outstanding presentation. On behalf of Bill Parker and I would like to present Dr. Gallardo a check for $100,000 in support of this research. Congratulations.
Thank you so much. I look forward to working together and also seeing the progress of all these outstanding projects that are being proposed.